So I will start my talk by discussing with you the progress that we made more in the field of the technology of genetics, and I will explain how we apply this in our research. And then my colleague, Professor Brunner, will talk more about the clinical impact of this work. So I will start briefly with some of the challenges that we face in medical genetics. I will talk about the technology that we use, whereby we can go from sequencing individual genes to sequencing the entire DNA of an individual. I'll illustrate how we can use this in our research to study the causes of disorders like intellectual disability, and I'll put a lot of effort on explaining to you the role of de novo mutations in disease. And finally, I will end with just a little bit looking forward to think where we can actually move our field further, because there are so many more applications where genomics can make an impact in medicine. So clearly, it starts with the patient in our field. And there's many questions that are encountered in medical genetics when parents have a child which is not developing well. They want to know what's happening in their child. They want to know what's the cause. They sometimes feel that they are perhaps guilty, something happened during the pregnancy. They want to know whether something can be done about it. And of course, they also want to know, if I get a second child, what's the chance that I get another child with a severe disorder? So they want to learn a lot of this. And for many of these disorders, for finding the genetic cause of these diseases, of course, we have to look at the DNA, at the genome. And our genome is very, very large. It consists of 6 billion nucleotides, and we have to study all of these. And we know also that people vary a lot, at about 4 million positions if you compare the genomes of two individuals. At the same time, these individuals are mostly healthy, and they vary already at 4 million positions, but we know that just a single nucleotide change can cause a severe disorder. So it's a very big challenge in that area. And this was really difficult in just a few decades ago, because we had technologies who did not allow us to look at everything. We had technologies like Sanger sequencing, whereby we could sequence individual genes. So we needed a clinician to help us, to guide us to the right gene to be tested. And then we had the microscope, where we could do chromosome studies, whereby we can look at the entire DNA, but we can look with a very low resolution. Later on, we replaced this with genomic microarrays, and we've been involved very much in using genomic microarrays to replace karyotyping. And this was very good, because you could detect smaller deletions and duplications in the genome, but still you could not detect the most small variations, the single nucleotide mutations. Now, a lot has happened in the last 10 years, and we have technology now which is called next generation sequencing, which is a bit strange because, of course, we already use it now. And it's really because of miniaturization and parallelization that we're now able to not sequence just a few DNA fragments at a time, but actually to sequence millions of fragments at the same time. And that has a very big impact on our field, because it allows us to go from an area where we actually needed an expert to say, okay, let's look at this gene or look at that gene, now to look at all genes at the same time, or even look at the entire genome. And that is something that is really changing our area, because it is really allowing us to look at the entire genome for the first time. There's lots of companies involved, there is big competition in the field, and this is fantastic, because that is driving the prices up, and it's raising the quality of reading the DNA. Because you have to realize that when you sequence the DNA, if it's very large, like a human genome, you have to have a very good measurement of the DNA, because else you will actually look at lots of mistakes, whereas that's not what you want if you would like to look at the individual causes of disease. So where we're now moving to is an area where we can look at the entire genome. And that is allowing us to look at all variation in just a single experiment. And that is, of course, very, very important in our research and in our clinical care. Clearly, as I mentioned, it's very important that the genome has to be measured very accurate and very fast, because we want to do clinical diagnosis in days instead of in years or in months. And it has to be affordable, and nowadays this is becoming a reality. The nice thing of genome sequencing is that you can actually have a single test for all disorders. So it doesn't matter what your patient has, whether he has intellectual disability or whether he has cancer. You can look with the same test at the genome of this individual. So that makes it in the laboratory very easy, because you have a single test. So you can do with every patient the same test, and you can then look at all the variation and try to interpret this. And clearly, this will be different for intellectual disability, where there is often just a single mutation that causes disease, whereas in cancer you see that it's a very big genomic instability because of the cancer process. So this figure is very important in our area. It is a figure that we use a lot, where we show that the price of sequencing a genome is dropping dramatically. And this is really something that's happened just since the beginning of this millennium, whereby sequencing a genome was still a few €100 million, and now we can sequence genomes for about 1,000 or 2,000 euros, which is, of course, something that makes the technology affordable in our clinical settings, which is great. And with that, I think our field is moving from an art whereby people had to be good at recognizing chromosomes, and it's really moving to to an industry whereby we do the genetic test in an industry scale, putting in the DNA of large numbers of patients at the same time, and then reading out the DNA in our computers and trying to interpret it and link the genetic variation to disease. So just one thought that sometimes people ask me, why would you perform genome sequencing? You can also do other kind of sequencing, testing a few genes, for example. And I think perhaps in 10 or 20 years we will look back at that and we think that this is a very strange question. We will say, well, this is about like asking the question, why not read an entire book, right? If you think about a genetic disorder, you want to look at the entire DNA and try to interpret this. We have to be very careful that we think already we know a lot about genetics, because we really still live, I think, in the dark ages of genetics. It may be that you see a certain mutation in one gene and you think this is the cause in your patient, but actually when you look at the genome you'll find that there is a big deletion somewhere else on another chromosome that is actually the cause of the disorder. So I think that is something that we have to all realize, that we're still at the start of our field. And of course there's key advantages of the technology. It's complete, so you have all variation, all different types of variation in the DNA, from the point mutation to the chromosome abnormality, and you have it in a single test. So it's also simple. That's very important. Because of this reason, genome sequencing centers are set up around the world. So this is becoming very big. And I know also here in Saudi Arabia, genome sequencing is being set up in large-scale centers. And this is very important. Not only the sequencing, but also the bioinformatics, the interpretation is very important to put forward. In England, they're sequencing 100,000 of genomes. In China, up to millions already. So this is becoming something that is not longer for very few patients. It's becoming a routine care in our clinic. And this means that a lot of the work is actually moving from, let's say, the bench, to the people doing the laboratory work, to actually the people doing the interpretation. And bioinformaticians are crucial in this field, and really high capacity computing is important. And we have to invest both in learning people to work with genetic data, as well as handling this and putting this forward again in a clinical practice. So with this as an introduction, I think we have to be honest and say that we are at the new start of genetics again. It's really fascinating to work in our field where technology is changing us, and it's changing the way we do business, it's changing the way we are diagnosing patients. And let's now apply this to a disorder. So we've been working a lot on the genetics of intellectual disability. And intellectual disability has been a little bit of a paradox, because, as you know, intellectual disability is an early onset disorder, and when it's severe, it actually has a big impact on fitness. So that means that if you have intellectual disability yourself, you will not likely get children, so that actually is reducing your fitness. At the same time, we know that intellectual disability is occurring very frequently in our population, in the Netherlands in about 1% to 2% of the population. And we know that intellectual disability has a high genetic component. So you see that in most cases, healthy parents get a child with intellectual disability. And people have been wondering, why are disorders with a reduced fitness still so frequently present in our population? You would imagine that they would actually be selected out. And one of the explanations for this, so this is actually not only happening for intellectual disability, it's also happening for many rare syndromes and also for the severe forms of autism and schizophrenia. So you see often a sporadic case, just a single patient in our family, and especially in the outbred populations you see this happening. In the Netherlands, we've looked at this in Nijmegen, 92% of our patients with severe intellectual disability, they occur sporadic, so you just have a single patient in your family. One of the explanations that you can think of while this is explaining why these disorders still occur in our population is actually that there is new mutations coming into the population. So you have a selection against the disorder once you have it, but there is new mutations giving rise to new patients in every generation into the population. And what's happening is you have to imagine it's just very simple. You have, of course, your DNA is being copied with every cell division, and when your sperm cells and oocytes are being made, this happening is also, and what you could happen is you could have copying mistakes whereby mutations are introduced into the sperm cell, which then can give rise to the next generation and thereby introduce new mutations in the DNA. So this is something that we are very interested in studying, and we know the role of de novo mutations in disease very much. Down syndrome with the trisomy 21 is explained by de novo mutations. It's a de novo chromosome 21. But also for very rare syndromes, we know that they are often explained by just a new mutation of a single nucleotide. So we know that these mutations, these de novo mutations, occur in rare disorders, and we know that they occur at all different levels of the genome, from the point mutation to the entire chromosome. So this is something to realize, but so far we had difficulty studying these. And now, because of the technology, we can for the first time look at the role of de novo mutations in the entire genome. So what we actually do here is we sequence not only the genome or the exome of the patient, but also the genome of the parents who are healthy themselves. And then in the computer we can actually look at the variation. We can see what is inherited from either the father or the mother, but also we can identify the new mutations that come into the patient, and that may explain the disease in the patient. So that is very important, the combination of sequencing an entire genome, but then also not doing this only in your patient, but also doing it in the parents. So we started with this in 2010, and we started not with genome sequencing, which at that moment was still too expensive. So we started with, let's say, an alternative, which is called exome sequencing. It's a technology whereby you sequence all the genes in the genome, but not the parts outside of the genes. So this is about 1% of the genome, so it's a little bit less, but it's still important because those are all the functional parts that we know of. So we started to sequence exomes in these patients and also in their parents. In the beginning, we did this for very rare syndromes, whereby we don't know so much yet about the genetic causes, and we have only selected a few patients. So, and the approach we took is we would sequence the exome of a few patients with the same phenotype, so the same syndrome, and we would look then for severe deleterious mutations in these patients, follow these up in the parents, and then follow up additional patients. And what we identified is that for many of these unexplained syndromes, exome sequencing allowed us to identify de novo mutations in genes explaining these rare syndromes. So this was very good for all these patients with these rare syndromes, and this was not only done by us, but by many groups around the world, and it's still happening, and this is allowing us to explain the rare diseases in many of these patients, which is great, because they really need help, and we can now diagnose them rapidly if we can find their genes. So here you see how many genes are now being discovered because of the use of exome sequencing and genome sequencing. This is important in the clinic, but of course also from a biological point of view to learn about these genes and their function and what we can do about them. So we wanted to take this further. We were wondering whether de novo mutations were only important in rare disease, but also perhaps important in common disorders. And we knew from the first studies done in genome sequencing of parents and their offspring that an average person has about something like 50 to 100 new mutations in their genome. So every one of us will have something like 50 to 100 new mutations in your genome. And that means that you have something like 1 to 2 of these mutations in the coding region. So 1 to 2 genes that you have will have a new mutation. So we were wondering whether these could cause also more common disorders. And for that you have to think about the mutational target. So it's actually shown here below. So what you see here is that for a very rare syndrome, Charge syndrome, we see that all the patients are explained by mutations in the CHD7 gene. So there's de novo mutations in this gene that explain that disorder. Well, fortunately, this is just a very small gene. This is just one gene, and because you just have 1 or 2 mutations in a gene in total in your genome, it means that the chance that you have a mutation in this gene is actually very low, and that is fortunate because it means that this disorder will always remain very rare in the population, occurring in about 1 to 10,000 patients. However, if you think about a disorder like intellectual disability or autism, we know that there is actually probably more than 1,000 genes that when just one of these genes is mutated, you can already get intellectual disability. So that means that the mutational target is much bigger and you have a higher chance that one of these mutations can cause disease. So this is what we wanted to test. And we did exome sequencing in patients, but also in parents of patients with intellectual disability. And we were looking for these kind of mutations here, whereby you see a zoom in of a next generation sequencing result. You see that everything in gray in this area is actually comparable to the normal genome. But here you see that there is a C instead of an A here. And you see that in the parents, this C is not present. So this means that there is a new mutation occurring in the patient, and it's occurring in the gene that actually is giving rise to intellectual disability. So we started in 2010 with the first 10 patients, and we identified already a few de novo mutations in genes that we could link to intellectual disability, but also we found many mutations that in genes that were not yet known to cause intellectual disability. We confirmed this in a larger study in 2012 with 100 patients, and at that time we actually already implemented exome sequencing in our diagnostic setting, and we could diagnose in the first study 16% of the patients with a de novo mutation. So we concluded that trio-based sequencing of the exome is very important for detecting these de novo mutations, and these de novo mutations appear to be a major cause of intellectual disability. And we also saw that exome sequencing is a good diagnostic test, so we implemented this in our diagnostics. Now we had very many new genes, and of course that is very important in our area. We realized that we know not so much about the genetics of intellectual disability, so we had to sequence many more patients in order to find a second or third patient with a mutation in the same gene. And we used very different approaches for this, and one of the most important actually turned out to be so-called targeted next-generation sequencing, whereby you take something like 50 to 100 new candidate genes, and you sequence them in 10,000 patients or so, and then you can find a second, a third, or a fourth patient with a mutation in the same gene, and then you can go to the clinic, you characterize these patients, you do functional studies to learn about the impact of the mutation, and thereby you can characterize and determine a new gene for intellectual disability. So this is something we do in great collaboration with groups around the world, including groups here in Saudi Arabia. And this is very important for our field because this is a graph showing you that the number of genes that we can now confidently link to intellectual disability is already more than 700, and it's really sharply rising because of all the new discoveries that we are making together. And that means that also the approaches that we use in order to diagnose these patients have to take this into account. So for intellectual disability, exome sequencing or genome sequencing is really preferred because you can actually update your analysis. But if you would make a targeted test, you would have to update it every year almost to include new genes that are being discovered so rapidly in your clinic. Now I just want to briefly talk a little bit about the move from exome sequencing to genome sequencing. In 2014 we published the first study about this, whereby we sequenced patients who were negative after exome sequencing, and we sent the DNA of these patients, again together with their parents, to a company called Complete Genomics, and they did the genome sequencing for us, and we looked at many of the things that we could detect in that. And one of the few examples that I want to show are interesting just to highlight the power of genome sequencing in the coming period. So one example I wanted to show is a patient which was seen in our clinic, and the patient fits with the Rett syndrome spectrum. And for this spectrum, actually the gene MECP2 is the likely causative gene. So our clinical geneticist had indicated that this gene should be tested, and it was tested by Sanger sequencing, and it turned out to be negative. But the clinician was he was very strong believer that this must be the truth, so we did an additional test. It's called MLPA, it's a quantitative test, and again it turned out to be negative for deletions in this gene. But finally we did genome sequencing, and we found out that indeed the clinician was right. There was a deletion in the MECP2 gene, which was missed by Sanger sequencing and by the MLPA test. And it turned out that this is the explanation. So the deletion takes out the primers which are used for Sanger sequencing, so you can only sequence the wild type, and the probe which was used for looking at deletions was located outside of the deletion. So I think this is a very good take-home message, that many people think that genome sequencing is important for looking at patients when you don't know what their probable cause is, but actually in this case the clinician knew already that it's likely to be in this gene, and actually it turned out that genome sequencing was the best test to detect this. Another very nice example that we identified in our first cohort of 50 patients was a duplication on chromosome 4. A duplication was missed by the standard microarray technologies that we used at that moment. But if we would have seen it with the microarray technology, we would have said, well, there is a duplication there. We would have looked at the genes in that genomic region, and we would have said, well, these genes do not explain the intellectual disability in our patient. And that's indeed correct, because these genes in this region do not explain the intellectual disability. But what we did is we looked further at the data, and it turned out that this duplicated sequence no longer was present on chromosome 4, but it was actually integrated on chromosome X. And when we looked at the data of our genome sequencing study, it turned out that on chromosome X, this new duplicated sequence was integrated into a gene, and it disrupted the reading frame of a gene on chromosome X called IQ-SEC2, here, this gene. And the name of this already gives it away. It's called IQ, and indeed this is a known intellectual disability gene. So what you have here is a duplication on chromosome 4, giving rise to disease on chromosome X by disrupting a gene located on this chromosome. And that's clearly an example where genome sequencing can only give you the answer. That's not something you can see by Sanger sequencing or microarray studies. So for this very small first study, we determined the diagnostic yield, and of course we knew that many tests were already done. And one of the things we wanted to know is what would be the diagnostic yield if we would have done genome sequencing as a first-tier test. And that's shown here at the bottom. You'll see that in our cohort in Nijmegen, we expect that we can now diagnose 62% of all patients with unexplained intellectual disability with an IQ below 50. And you see that in our cohort, most of these are caused by de novo point mutations, and then we have de novo copy number variations, and we have only a very small part where there is recessive inheritance. Also, you see that there is still a very large part which is unexplained, and that is because there may be many things, like for example non-coding mutations, that so far we have not been able to interpret in our clinic, so we still have a long way to go. The good thing, however, if you look at this from just looking a bit historical, you see that let's say the power of this genome technology, as well as our increased knowledge on studying the intellectual disability genetics, has allowed us to diagnose now more than half of our patients with severe intellectual disability. If you just look back something like 20 years ago, we could diagnose less than 10–20%. So this is good news, and of course this is a starting point also for further development of therapies. So I wanted to conclude and then just have one more brief outlook to the future. So to conclude from our work so far is that next generation sequencing technology is really allowing us to look at all the genes or even the entire genome of a patient. I think that we will all conclude that as soon as it's affordable, genome sequencing is the best technology to use for detecting and mapping both de novo point mutations as well as structural genomic variations, as well of course as all the inherited variations that every genome harbors. De novo mutations in genes already explain the majority of severe intellectual disability in outbred populations. Of course, in inbred populations there will be a large recessive component as well. And there is many questions that we want to study further on the role of de novo mutations, including studying the role of non-coding de novo mutations, the risk factors. One of the risk factors that we identified, and others as well, is that actually an aging father is the biggest risk factor. So most of these mutations, about 80%, occur on the paternal allele, and older men, they give more mutations to their offspring. So this is when I always tell ladies to think about their future husband. And then finally, of course, I also want to look at other disorders which can be explained by de novo mutations. Just one slide with a look at, and then I will end. I think there is a whole lot to be done for genomics in the clinic in the coming period. And it's great to see so many specialties here present who are interested in genetics, because indeed it's important for many disorders. Genome diagnostics really will become a standard and a first-tier test for most genetic diseases, I'm sure of. There is many of us also working in things like non-invasive prenatal genetic diagnostics, so that is a very important field of research. Of course, for recessive disorders, we can do carrier testing of a genome of future parents. We can develop pharmacogenomics tests to prescribe the right drug to the right person. Especially in cancer genome sequencing, of course, this is important to tailor the treatment. And of course, we will also move to an area whereby perhaps you and me will have our genome sequenced just to look at our genome and think whether we can perhaps prevent disease from occurring. The question really is how far we want to go, and that's something we have to discuss together in our field, but also, of course, with society. I want to thank many, many, many people. I've decided here to put many of my colleagues from Nijmegen and Maastricht who've been really fantastically in my work. I cannot thank all of them enough because this is work from a big team. And of course, I want to thank Han Brunner, my colleague who I've worked with so much pleasure for so many years, and I will continue to do that. And I want to thank all the patients and families involved and all collaborators around the world, as well, of course, as some of the financing. Thank you very much.
So simple questions never have simple answers. Here's the expert.
Yes. This is always nice when you get a question like this. No, so I think it's, of course, this technology is tremendously important, and it's only just happening. So we don't know the impact of this technology on gene therapy as a whole. I think it's important, it's more or less it's a combination. So we need the genome sequencing for diagnosing the patient, and then we need to see where we can do something. Now the problem, of course, is that many of these disorders, like for example intellectual disability, where the mutation is already, let's say, having an effect during the development of the child, it will be very difficult to treat, even with the new technologies like the CRISPR-Cas. So I'm not immediately sure we will, immediately benefit for all these rare disorders, but especially disorders which occur a little bit later or develop a little bit later. Perhaps some things like blindness, where it develops during, let's say, during in the years. There, indeed, these kind of technologies will have an impact, and of course, the idea that you can specifically target genes and correct mutations is fascinating. We are both not experts on the therapy field, but we are investing in these kind of technologies, and we have colleagues working on this. Yeah, no, so I think it's absolutely true that let's say the diagnosis can be very different in countries like here, where you have multiple affected. I mean, I think in general, just an answer, in general, when people ask to me like targeted versus, for example, exome, or do first targeted sequencing and then do exome sequencing, I think, yes, there is a possibility for this, and definitely in this period still is also economically perhaps more feasible to do the targeted sequencing. On the other hand, if you look, let's say, 5 years ahead, we see already that we can do now exome sequencing for a few €100, and I expect that we can do genomes for a few €100 in a few years. And then I think this discussion on doing, let's say, targeted first before you do a genome, I think it's more a discussion on let's say, the ethical discussions, what do we want, the incidental findings. Then it's an economical, let's say, discussion, and let's say how to stratify. So in that way, we've said we will move forward to the exome and the genome because we think that is where our field is moving. It's more expensive at this moment, but actually it will help us to make this transition. And that's also perhaps our role in the field, to try these new technologies. But of course, many laboratories around the world currently are using these targeted panels, and I think especially also, like you say, this is very important.